All right, so we're back. Sorry, so I just started recording the lecture. Now we're talking about gene expression as a review first. And as I said, this gene expression can be sort of understood as what we call a central dogma of biology. Central dogma of biology. So again, this is something that we've learned before, but the main idea behind the central dogma, I'll just make this a little bit prettier here today, is that you have DNA, deoxyribonucleic acid, which contains what we call genetic information on how to build these proteins inside your body. But it's going to actually go through a couple of steps. That DNA is going to be transcribed into what we call messenger RNA, which is translated into what we call proteins. And of course, this may be made up of one or more polypeptide. So I'm going to write polypeptide here as well, just to kind of keep things consistent here. All right, so we have DNA to RNA to protein. And the central dogma of biology that you see here is actually pretty complex, right? Because we can break it down into the ideas of transcription in the first part and its mechanisms, and we can also talk about translation. Now, again, I don't want to go too deep into this, but just to give you a brief example of some of the things that you have to know is that, first of all, DNA, right? DNA as a molecule exists as an anti-parallel double helix that looks something like this, right? So hopefully you guys remember this. Now, the anti-parallel aspect of this is that one of these strands goes from 5' to, let's just make sure that I'm doing it the right way. Yeah, 3' there. And the other strand is going from 3' to 5', just like that. And of course, the backbone that you see here is going to be what we call the phosphosugar backbone, right? So it's got a ribose sugar and a phosphate. And of course, in the middle, we have what we call our nitrogenous bases. I'm not going to write nitrogenous, let's just write bases here, which are built into this sort of adenine to thymine and guanine to cytosine relationship, wherein the hydrogen bonds that exist between each one of those relationships are a little bit different. So A and T have 2 hydrogen bonds, and G and C are going to have 3 hydrogen bonds. Okay, great. So that's what we have going on. But the thing about DNA is that we can actually take this entire model that we've drawn as a double helix and sort of write it out a little bit differently. We can write something like A, T, G, C, C, G, C, G, C, and let's write T, G, A like this. And of course, on the opposite side, let me write T, A, C, G, G, C, G, C, G, and I think it's going to be A, C, T over here with the 5' end right there. And this is a perfectly valid way of expressing genetic information. No problem about that. The thing that we have to understand is that, you know, when you take a look at DNA, there are actually 4 different ways in which you could potentially read this DNA. You can read from 5' to 3' this way and have that conveying information like ATG, CCG, and so forth. Or you could read it this way, like TCA, GCG, and so forth. Alternatively, you can read from here to here, TAC, GGC, or AGT, CGC. And all of those are potential directions and potential ways in which we could read this DNA. However, I hope you guys remember that anytime we deal with DNA or RNA, we're only going to be encoding information from a 5' to 3' direction. So I'm going to get rid of these 2 arrows here that are never going to be feasible ways in which we read DNA. Now, of course, as long as you're reading from 5 to 3, you're okay, right? That's the way that information is going to be encoded. So either one of these would work, but for the purpose of this example, I'm just going to say that what we have up at the top here, the top strand, from ATG, CCG, CGC, TGA, that's going to be the information that is coded in this DNA. And that information, of course, is the information that is going to be used to create a polypeptide. So if I were to say that this top strand right here is in fact the one that's going to contain information, typically we would refer to this as the coding strand. The coding strand as in the strand that is used to make some sense of what that amino acid sequence is going to be. Whereas the opposite strand from 3 to 5 that goes TAC, GGC, that's going to be called the template strand. And that's because if you want to create an mRNA molecule, which is what we were trying to do here in transcription, you're going to be using the opposite basis to what you want so that the complementary basis can actually give you a brand new mRNA transcript that is going to reflect what the coding strand was going to be. Now, as you guys remember, I like to think of this as a stamp. If you want to have OK stamped on your paper, your stamp is going to look something like this, and then that's going to flip over and stamp the right information. So that's the same type of what we call template base that we're trying to describe here. But in any case, we haven't really gotten there yet. So for now, let's just take it for granted that DNA has multiple directions in which they could be read, but only one of them is going to make any sense at all. And this is going to be the coding strand, and that's the template strand here. And that's what DNA is going to look like. Now, of course, when we go to RNA, when we go to creating RNA molecules, you'll notice that RNA and DNA are actually very similar, except just a couple of differences, right? For instance, A is going to be A, but T is going to be U. G, CCG, CGC, UGA. So you can see that this RNA is a copy of this DNA right here, except we're switching up some words, like some letters, like T for U, and of course, the sugar, the ribose sugar here is a little bit different. We don't have a deoxyribose sugar, but that's fine. And this process right here, called transcription, happens as a result of a fairly complex mechanism that we'll just briefly talk about right now. All right, so let's suppose that we're looking at a chromosome, and this chromosome has this long stretch of DNA. Now, along the stretch of DNA, we might have a gene. So let's call that a gene. Now, this gene contains the right information in its DNA. And of course, there's 5 to 3 and 3 to 5 and all that inside here, because this is actually DNA, right? It looks like this. So this gene might have information, but we also want to let the cells know that, look, here's a gene worthwhile making. Here's a gene that needs to be polymerized. So the way that we do this is by having what we call a promoter that is somewhat upstream of the gene itself. And typically in the eukaryotic cell, this promoter is going to have what we call a TATA box. So it's going to be called a TATA box for the fact that it has TA, TA, AAA that allows this, allows this region of the gene, allows this region of the DNA to interact with what we call transcription factors. All right, so the whole point that we want to try to do is to take this DNA and we want to create a messenger RNA, or at least a pre-mRNA. So the idea here is that this promoter is going to allow several transcription factors to sort of bind. So here's TF, here's TF. What that does is allows RNA polymerase right there. So that's the RNA polymerase to bind. And this RNA polymerase is going to start copying that DNA into a new strand of RNA that kind of snakes out as it moves forward. So hopefully you guys remember what this is talking about, right? Because if we were to sort of zoom in on what's really happening is that we have, let's say, the polymerase like this, and the DNA is actually split like this. So let's see. Split like this. And it's usually going to be about, I don't know, let's say 10 nucleotides at a time. 10 nucleotides at a time. So if I have 5 to 3 prime here, this would be 3 to 5 prime here. And the new RNA is actually being produced this way, right? It's being produced in this direction right there. So this is going to be the coding strand. This is going to be the template strand. And the new RNA that we're making is going to be complementary to this template, which is going to be identical to the coding strand right there, minus T's being used. Okay, so that's what's going to happen here. And eventually this falls off, which then produces what we call a primary transcript, or sometimes called pre-mRNA. So hopefully you guys remember these mechanisms. I'm not going into details about these mechanisms, but hopefully you remember them, because mastering these ideas is really going to help you understand the next section a little bit better. Okay, now the next thing that you guys have to know is, of course, before this pre-mRNA becomes a mature messenger RNA, it undergoes 3 different modifications. So 3 what we call RNA processing, things that you guys probably need to review. Now, the first one here is going to be called the 5' cap. okay, 5' cap. So this, imagining that this primary transcript is 5 to 3' right there, this 5' cap is going to add right here to the 5' a methylated GTP cap, right? So this is called the methylated GTP cap. And the purpose of this cap is to protect this messenger RNA as it enters the cytoplasm. Because remember, all of this is happening inside the comforts of a nucleus, right? So this is a nucleus. But outside the nucleus, in the cytoplasm, this is a very dangerous place to be. You know, we have this nuclear pores that can take you there, but once you go out, you know, we have all of these hydrolytic enzymes. There's these enzymes that want to actually break the mRNA down. So we want to make sure that we protect ourselves at the 5' end right there, and that's going to be the 5' cap for protection. Now, the second thing that we do is going to be what we call polyadenylation. So this polyadenylation is going to happen at the 3' end, and as you can see here at the 3' end, the polyA signal, which is usually AAUAA, is going to actually just add a bunch more A's, a ton of A's to the end of the 3' end, rather, of this particular mRNA. Now, the reason for this, again, is the same as the reason for this, is to protect what we have within the actual gene encoding region of the mRNA to be preserved and not destroyed. However, one thing that you do notice is that while this 5' cap here is literally a cap that makes that entire region of the mRNA unreactive, the poly-A tail just seems to be this random length of tail that sort of simply degrades over time, just like this, in the cell. So you might think, well, that's kind of bad. Why not just put a 3' cap as well? Well, but the reason is that this set of A's, all of these A's, is actually able to put a bit of a timer on the mRNA. So for example, let's suppose that this mRNA comes out into cytoplasm and it has, I don't know, this many mRNA, this many A's, right? So remember, all of these enzymes are coming by and they're cleaving that off, boom, right? And then they might cleave this off, boom, and cleave this off, boom, cleave this off, boom. Now all the A's are gone, but because the A was short, maybe that might have taken 20 seconds, maybe it would have taken 5 seconds, and so on. But if the cell really wants this mRNA to exist in the cytoplasm for a very long time, for instance, we're talking about a very sustained production of a particular protein, then the cell would opt to create a much longer poly-A tail, in which case it's just going to be that much longer before the hydrolytic enzymes start getting to some of the some of the gene coding elements of our mRNA. Does that make sense to you guys? So that's the two things that we've already talked about. Okay, now the last one, of course, is a little bit more interesting. I think you guys might find this more fascinating. It's called, oh, by the way, this section right now that we're talking about, of course, deals with a little bit of evolution, but here, it's going to be called RNA splicing. RNA splicing. So let me just sort of redraw that gene in a little bit more detail though, right? Because we have the gene that looks something like this. And what we'll see is that some of these gene regions are what we call exons. Like that one, that one, that one. I don't know why I put plural there. Let's get rid of that. Exon. And some of these other guys are what we call introns. So here in this case, we have 2 introns. Now, the thing about RNA splicing is that this only happens in eukaryotic cells. It only happens in eukaryotic cells. It does not happen in prokaryotic cells. And what that means is that having these introns here are going to be actually genetically advantageous. And we sort of explained why this might be the case with these exons and introns and exons recombining together to form a single protein. So we can say protein 1, and maybe all 3 of them will create protein 2, and so on. So because you can pick and choose which exons you're going to actually leave behind and which intron you're going to leave behind and which exon you're going to add to the overall translated portion, you're able to actually create a lot of different combinations from these exons to be able to create additional proteins from a single gene. Because here's the idea, right? Humans have 22,000 genes, but we have way more than 22,000 different proteins. So if we assume that a single gene product is going to make a single polypeptide, which then falls into a single protein, right? Then there's no way we'll have way more than 22,000 proteins if we only have 22,000 genes. So in some ways, what this is doing is it's actually acting like a data compression system, right? You have the same amount of data, but you can actually modify during decoding that data that allows you to actually have greater amount of information hidden within a smaller set of information. And as you guys can tell, the idea of ensuring that you don't have so many genes, that your genome gets so big. I mean, the main idea there, of course, is that the bigger the genome, the longer it's going to take to copy, the more mistakes it's going to accumulate. So this RNA splicing is allowing you to, first of all, keep that genome as efficient as possible, as small as possible, with all the parameters that are required for that organism, and that will allow it to be more competitive in nature, right? You can reproduce faster and so forth. So that's what we have for RNA processing. So hopefully you guys don't have any questions on that front, yeah?
Mm-hmm.
All right, so now what happens to this? This comes out through the nuclear pore. So here we have the nuclear pore. And we're going to look at translation then, right? Okay, so let's take a look at translation in a nutshell. Now, translation happens in the cytoplasm for the most part, but it always starts on the cytoplasm anyway. And it starts off with the 5' cap of an mRNA and a 3' poly A tail here with all of these information that's in the middle. Now, as you guys probably remember, we have to remember that during translation, we're going to be using something called a triplet codon system, right? Triplet codon. So we've been talking about this extensively today, so I'm sure I don't need to review, but it takes 3 nitrogenous bases to encode for a single amino acid. So just going back to what we have earlier, we had AUG, CCG, CGC, UGA, and then UAA, something like this. And here's the poly A tail that looks like this. And then there's a lot of untranslated region here as well. All right, so we have a pretty good model of what this mRNA is. Now, the thing that you guys have to remember is that there's a very special way in which we translate what we call nucleic acid information to amino acid information, which is supposed to be a string of amino acids like this, right? So how do we get from the AUGC to the 20 amino acids? Well, that had to do with the laws of probability or combinations. For example, if A meant alanine, which is an amino acid, and B meant, I don't know, let's say cysteine, I don't know why, but and F meant phenylalanine, for example. Well, not F, I'm sorry. Hold on, I completely screwed this up, sorry. If A meant alanine and G meant, glutamic acid, and C meant cysteine, I don't know what the hell I was talking about, and AGCT meant tryptophan or something like that, then you quickly realize that we run out of the amount of information that we can use, right? So again, those are only 4 amino acids. We need to encode for 20. So this 4 to the power of 1 system doesn't work. 4 to the power of 2 system doesn't work. 4 to the power of 3 system, now it works. So we have those 64 codons, right? So hopefully you guys remember not like what the codons are going to be, but why that codon system is in existence. Sorry, I don't know why I started running A, B, C there. Okay, now, so let's actually get to creating these amino acid chains here. And we'll start off with the small subunit, as you guys remember, small subunit of the ribosome that attaches near the 5' cap. Now, after it attaches the 5' cap, it starts to move down the mRNA until it finds AUG, which is our start codon, right? It starts to move down until it finds AUG, and when it does, it will actually stop like this, okay? And it will wait for something. Now, what's it waiting for? It's waiting for what we call our tRNA. Now, I'm gonna erase this a little bit. tRNA, as you guys remember, is gonna be the direct link between nucleic acid information and the amino acid information. Because tRNA looks something like this. It has what we call the anticodon loop, which actually interacts with the mRNA. And because AUG, okay, because AUG is for methionine from that codon chart, this is going to actually have methionine attached to that tRNA. And so that's what's directly linking the AUG information that is present on the mRNA to the methionine, which is the amino acid that it actually represents. And the same thing with CCG here, right? So we're gonna go right there and put GGC, that's the anticodon. And this one happens to be proline, because CCG goes with proline. But let's not jump ahead there for a minute. So first of all, we get that tRNA to come in. Now the second thing that happens is that the large subunit will come in like this. Now today I'm not going to write the details of the E site and everything, but this is going to be the P site here, and this is going to be the A site. Now into that A site is going to be that tRNA I just mentioned, right? GGC with proline. Now, once that proline containing tRNA is put into the A site, then in the P site here, we're going to drive a chemical reaction that disconnects that tRNA from methionine, just like that. And then we're going to transfer that bond over to proline right there. So you can see now we have methionine and proline that are connected together. Now, once this happens, we're going to have the small subunit move 3 steps to the right. So that is to say that if this moves 3 steps to the right, let me see if I can make this a little bit cleaner. We're going to have this ribosome sit right around here, just like that. And then the top subunit, or the large subunit, is also going to move 3 steps to the right. But if that happens, you'll notice that this portion right here is going to become the P site. And this one sort of goes away. And then, of course, this one becomes the A site. And it looks something like this, right? That's how it moves 3 steps to the right. So like I said, I'm not going to draw the E site here, but you could imagine that this tRNA now, the UAC tRNA, is going to sort of exit out so that it can be recharged. And a new amino acid or acyl tRNA is going to come in this way. So I wish I would have done this a little bit better here. So that's going to be CGC there. Okay, and of course, CGC was arginine. And arginine is going to be connected to a tRNA that has the anticodon GCG. So let's write ARG there. And then same thing. We're going to hydrolyze that bond and connect that proline to arginine right there. And as you can see, we just continue doing this until we finally get to UGA, which is going to be the stop codon. And at that point, instead of a amino acyl tRNA coming in, you're going to have a release factor that comes in and sort of dissociates everything. So that's basically the review of our central dogma of biology. Sorry, the last part here is a little bit confusing, and I try to go through it a little bit too quickly. But we've actually done this lecture before, so hopefully you guys will remember all of this. Do you guys remember all this from before?
Yes.
Yeah, you do. Okay, good. All right, now we get to kind of kick it up a notch. So let's do something fun now, okay? Because we're going to now talk about gene regulation. Because as you can imagine, what we've talked about up to this point is how DNA gets transcribed and then the RNA gets translated. But you guys have to understand that that's not going to happen for all the genes in all the cells. Like, for example, as I said, we have 22,000 genes in our genome, and we have a lot of different cells in our body. Not every cell is going to be producing every 22,000 of those genes. For example, if you're a stomach cell and the gastric pit cell, you produce, these proton pumps that ultimately produce hydrochloric acid, right? Now, that's because you're a gastric pit cell. But if you're a hair follicle cell, for instance, you're not going to be expressing that same hydrogen pump or that proton pump, because if you do, then you might be sweating acid, right? You're sweating hydrochloric acid from your face. So that doesn't make any sense. So depending on the purpose of the cell and even some things like timing, when and how much, Like all of these genes have to be regulated in order to perform the functions that the body is going to need in an efficient way. But the only problem is that when it comes to gene regulation, eukaryotic gene regulation is extremely complicated, okay? So gene regulation, that's what we're talking about right now. Eukaryotic regulation is extremely complicated, and that's why, for the most part, AP Biology doesn't really ask you too many questions, if at all, about eukaryotic gene regulation. But what they do want you to know are some simple models that we see in prokaryotic organisms, so that we're able to understand the basics of how genes can be regulated within the context of biology. So that's what we're going to do today. We're going to be looking at a simple prokaryotic model, maybe E. coli, and see what kind of mechanisms they have in place in order to regulate gene expression. So let's begin by talking a little bit about a few important issues. So let me draw that E. coli cell here. All right. So we're lagging a little bit, but that's okay. All right, so here's E. coli. As you guys know, it's a prokaryotic organism. It has a circular DNA, right? Because it's a prokaryote. It's got a circular DNA. That's the chromosome. Not circular DNA, but it's got a circular chromosome. Now, as you probably are aware, along this chromosome, there are a bunch of genes, right? There's a bunch of genes along this chromosome, and that's fine. But let's talk about what this E. coli eats for a moment. It's a heterotrophic organism, as in it doesn't have the capacity to photosynthesize, obviously. And what you have here is the ability for E. coli to uptake, I don't know, let's say glucose from the environment and actually undergo glycolysis. So obviously glucose is going to be one of the main food sources for any bacteria, especially things like E. coli. So glucose is a good option. But they don't just eat glucose. They could also consume what we call lactose. I don't know if you guys remember what lactose is, but lactose is in fact a disaccharide. So it looks something like this, and looks something like this. And this is going to be made from 1 part glucose and 1 part galactose, like this. Okay, so this could also be consumed and processed by E. coli as well. All right, but if I were to ask you guys, if the bacteria had a choice, if, let's say, E. coli was in front of these two different sugars and said, what should I eat first? What do I prefer? What do you think it would be? Do you think it would prefer glucose, or do you think it would prefer lactose?
Glucose.
Yeah, but why is that, Daniel? Why would it prefer glucose instead of lactose?
So lactose are hard to digest.
Yeah, I absolutely agree with you, you know, and we can be a little bit more specific about that, right? Because the thing is, if we want to digest lactose right here, what we have to do is we have to actually cut lactose in half first so that we can free up that glucose, and then we have to further modify galactose into something that looks like glucose so that we can put it into glycolysis. So you can imagine that glucose is going to be much preferred, and lactose is going to be secondary. Now, but looking at lactose here, this cutting of lactose has to happen via an enzyme, right? Via an enzyme, because remember, whenever we have a chemical reaction, we probably need some sort of a catalyzer, and that enzyme is going to, a catalyst, and that enzyme is going to be called, as you know, lactase. Colloquially, it's called lactase. Now, specifically in E. coli biology, we call lactase beta galactosidase. Beta galactosidase. Okay, that's what we call it. But you know, that's just a fancy term to describe what lactose is in bacteria. Now, the reason why we're talking about is simple. See, the thing is, if lactose is not present in the environment, then there's absolutely no reason that E. coli needs to produce this protein lactase. So this gene that it has, right? So lactase gene might be right here, right? The gene that it has for this lactase gene or lactase protein is going to have to be, let's say, off for the most part, and only to be turned on when we need it to be turned on, when lactose is present. Does that make sense? So we're going to be looking at a system wherein we have a switch that can turn on or off the genes as the environmental condition changes. So that's what we're going to be doing today. So let me introduce you guys to a new term, and the new term is going to be something called an operon. Operon, okay? So operon is going to be a group of genes controlled by a single promoter and an operator. Okay, and I'm going to draw out what this operon is going to look like here, because today we're going to be focusing on something called the Lac Operon. Okay, we're going to be focusing on this example of the Lac Operon. So let's draw out what this Lac Operon is going to look like here for us today. Let me see if I can draw this nicely. So here we have a, that's not bad. Here we have, okay, there we go. So here we have a chromosome, right? We have a section of the chromosome. So what we're looking at right there, maybe, you know, just this area of the chromosome right there from the bacteria. So it's kind of like that, right? That's what we're looking at. All right. And what we have is going to be a promoter that sits right there. As you guys know, the promoter is what interacts with transcription factors and so on. And then we actually have what we'll call an operator region right there, an operator. And then in this case, we actually have a cluster of 3 genes that we call the Lac Z, Lac Y, and the Lac A. And these are all genes that pertain to digestion or the metabolism of lactose. All right, that's what we have here in the Lac Operon. So now let's take a look at what each one of these genes actually do for a moment. So you know what the promoter and the operator, well, we haven't talked about the operator, but you know what the promoter does. Let's look at the Lac Z gene right here. So Lac Z gene is going to be the gene that produces beta galactosidase. So this is the lactose, this is the lactase gene right there. So that's fine. Lac Y produces an enzyme that we call permease, and permease makes it more likely for lactose to be brought into the cell. So you can clearly see that these are working in concert with beta galactosidase to bring in lactose, and then beta galactosidase cuts it out. And Lac A is going to be called a transacetylase. And we're not really 100% sure what it does, but from the name, it seems to me that it's able to transfer an acyl to a group, and I'm guessing that it's going to be involved in the processing of either galactose or, I don't know, or lactose itself. So we're looking at 3 separate genes that are all working together for the metabolism of lactose, which is a sugar that we're talking about near E. coli. All right, but in order to understand that this is controlled by a promoter and an operator, I actually have to introduce you to a different gene that is actually somewhere else inside the chromosome of bacteria. And for that gene, we also have a promoter, but this gene is going to be called a Lac I gene, Lac I. Okay, so what does the Lac I gene produce? It produces what we call the Lac inhibitor protein. So let's see what this inhibitor looks like. Now, first and foremost, inhibitor protein is going to have a 3-dimensional shape, and let's say that this 3-dimensional shape looks something like this. Actually, let's do it the other way. Let's say that it looks something like this. Okay, so that's what the Lac inhibitor protein looks like. All right, now when this Lac inhibitor protein is produced, what it's going to do is it's going to naturally have an affinity to the operator region right there. And if I want to just kind of make this a little bit more interesting, what I can do is just make the operator stick out like this. And you can imagine that this thing right here, whoops, is going to literally sit like this on the operator, just like this. It's going to sit there. Okay, why? Because it's an inhibitor. It's sitting on the operator, inhibiting the polymerase from moving forward. Now, let me show you what I'm talking about. Remember that we don't want this gene to be on when there's no lactose. So the given condition, the default condition is that there's no lactose. And when there's no lactose, these inhibitor proteins are constantly produced, and they literally sit on that operator so that the polymerase that lands here, can't move forward. They're blocked by this inhibitor protein. Does that make sense to you guys so far?
Yeah.
Okay. Now, when do we want to turn this gene on? or these sets of genes on. We want to turn them on when lactose is present, right? We want to turn these on when lactose is present. So here's what's interesting. Lactose, when it is present, as it were, has an affinity to the inhibitor as well. You see this little site right there at the top. This site at the top will actually bind to lactose, just like this. And when lactose binds to that inhibitor, it changes the conformational shape of this inhibitor and it falls off. So when lactose is inside the cell, then the lactose binds to the inhibitor, has it fall off the operator, and therefore the polymerase can now go past all of these genes and produce these enzymes that are needed to break down lactose. Does that make sense to you guys?
Yeah.
So it's a little bit confusing, but the whole point is that this inhibitor is going to be turned on when there's no lactose attached to it, and it's going to turn off like this when lactose is attached. When lactose is attached. So that means when lactose is available in the cell, the inhibitor turns off, falls off the operator, and therefore allows for that polymerase to attach to the promoter and push its way down and produce beta-galactosidase, permease, and transacetylase in working to break down the lactose that are available. So, you know, ultimately, we call this type of an operon an inducible operon. So think about what that means for a moment. Inducible operon. Inducible means we can turn it on. We can switch this whole thing on. That's what inducible means. So it means that it's off by default, and it's turned on when needed. And that's why for the Lac Operon, it's off by default because usually there's plenty of glucose available. But it's only when glucose is gone and a lot of lactose starts to come into the cell that these little inhibitors fall off of the operator, allowing the genes to be turned on for that purpose, for the purpose of breaking down lactose. Okay, good. So hopefully you guys understand what this is all about. Yes?
Yeah.
Okay, good. Now, what I want to show you now is another operon, but for that one, I'm not going to do too many drawings. For that one, I'm actually going to show you a slide or a set of slide here. So this is what we're looking at right now. This is actually chapter 18. Can you guys see my slides right now or no?
Yeah.
Yeah, there should be a little green thing that's showing up, but I don't actually see it. Yeah, I guess it's fine. You guys can see this slide, right? Overview, conducting genetic orchestra.
Uh-huh.
Okay, good. All right, so let me actually show you the Lac Operon first, okay? Because it's this one right here. So as you guys can see here, we have Lac Z, Lac Y, Lac A. There's the operator region and of course the promoter. And the Lac I, or the inhibitor, right? The inhibitor protein is usually bound to the operator given that there's no lactose. But when lactose is present, lactose is going to bind to the inhibitor right there, therefore having it fall off of the operator. And what that means is that the RNA polymerase can now go all the way down here and produce these three enzymes. So that's the Lac Operon that we've already talked about. However, the one that I want to show you now is going to be what we call a repressible operon. Repressible operon. It's called a trip operon. So let's take a look at this one quickly. So this is a little bit of background about why we might need the trip operon. In this case, we have a metabolic pathway wherein we begin with a precursor, and through this one enzyme, it turns into another substrate, and then through enzyme 2, it turns into a different substrate, and through enzyme 3, it finally turns into tryptophan. So it seems to me that, you know, tryptophan is an amino acid. So it seems to me that this is an amino acid that your body can actually synthesize using other components, whatever this precursor might be. So that's a good thing. So it's what we call a non-essential amino acid. However, one thing that you guys got to remember is that in the body, you always have to maintain some level of homeostasis. As in, you don't want to be doing this too much, to such a degree that you have way too much tryptophan and perhaps not enough precursor. So what you want to do is, if you have way too much tryptophan, to slow down this entire process so that you can bring down the levels of tryptophan and bring up the levels of the precursor and so on again. So in order to do this, we're also going to use some sort of an operon. So let's take a look at what that operon is going to look like. And this is what it looks like. It's what we call the trip operon. Now this looks a lot more confusing than it actually is. So let me explain what we're dealing with. First of all, let's take a look at the trip operon. We have a promoter, we have an operator. But in this case, the operator is open by default. So this is on by default, not like Lac Operon that was off by default. This was on by default. So it always allows for the transcription of this gene EDCBNA. And when those genes get transcribed, they produce EDCBA proteins. And as you can imagine, going back here, those EDCBA proteins end up creating these enzymes that are part of that metabolic pathway. So when this operon is turned on, or on by default, we're constantly making enzymes that facilitate the transformation of that precursor into tryptophan. Now, somewhere else along the chromosome, yet again, here we have another thing called the TrpR gene, which is going to be a regulatory or repressor gene over here. And the repressor gene is going to produce the repressor. But unlike the Lac inhibitor, this repressor is off by default. As you guys can see, it says it's inactive. So when it's created, it's inactive. So this doesn't really do much, all right, at the moment. So we get all of these enzymes being produced. We get a ton of tryptophan, no problem whatsoever. But here's the thing. We want to slow this down when there's too much tryptophan. And there's a really easy way to solve this. Now, if you go back to this inactive repressor, let's look at it right here. All we have to do is make this repressor sensitive to tryptophan to such an extent that when tryptophan is too much, it binds to that inactive repressor and turns it on to an active repressor, which can now go down to the operator and boom, block the polymerase from moving forward. So in this case, what we're doing is that everything is on by default, but when the product concentration becomes too high, that concentration feeds back into the tryptophan operon trip repressor, and that allows for the repressor to become activated and bind to the operator so that, as you can see here, this RNA polymerase can no longer create these EDCBA genes. Now, again, you know, this would actually result in the slow production of tryptophan to such an extent that we're not going to get a lot of tryptophan anymore because those enzymes are no longer being created. Now, what that simply means is, you know, we're going to start using the tryptophan we do have available inside the cell. And at some point, even this tryptophan is going to be broken down because we have no other tryptophans that we can break down or we can use anymore. So we break this tryptophan off, or it just falls off. We use that tryptophan, and obviously at this point, we probably need more tryptophan again. So that's good, because when we cleave this off here, it falls off the operator. And once again, these EDCBA genes are again being expressed, and they're able to create more tryptophan. So it kind of creates somewhat of a homeostatic, sort of a sinusoidal graph, right? So you have, I don't know, tryptophan levels on the y-axis, time on the x-axis, and you have this sort of a, you know, rise in tryptophan levels. These operons get turned off, so it goes down. When it's low, the operon gets turned on, so it goes up again, and it sort of cycles like this over time, you know, enabling the maintenance of homeostasis. So is that pretty clear with you guys what the trip operon, the Lac operons are?
Yeah.
Okay, so it's pretty simple. It's not too difficult, but you do kind of have to think a little bit about, you know, what's turning something on, what's being turned off. So these are just different ideas, but that's pretty much it for gene regulation. I mean, there are some other eukaryotic regulation stuff that we could potentially see, but I highly doubt it. So I'm not going to worry too much about eukaryotic gene regulation at this time. We will go over that if I think we need to with new information that's coming out on April 4th. But that's really it for gene expression and regulation. So what we have left for the next hour then is biotechnology. Okay, and we want to talk about a few things that are really important. So what we should do is just take a brief break now and start again around maybe 7:03 or so. I know all of your clocks might be different, so you can take a brief bathroom break or whatever. I'm going to make myself some tea. So let's come back and continue our lecture, okay? All right, I'll be back. Yeah, what's up? Sounds good? Okay, cool. All right, I'll be right back. Hi guys, are you guys there?
Yep.
Okay,
Yeah.
okay, good. I just got a message from our district office of education, and it says that there's a 영업 중단 권고 및 감염 예방 수칙 준수 명령. I think, oh boy,
What's that?
I don't know. I think, I don't know. I think, oh, I think there's some things that we have to do. Okay, never mind. We don't have to close. Okay, hold on. Let me just get this call. 네, 정희쌤. 네. 네네. 네네네. 아, 네. 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 음. 아, 그런 거. 오케이, 오케이. 그러면 지금 뭐 이거 뭐 다들 하는 그런 경보는 아니고요. 아, 진짜 미치겠어. 얘네들 왜 이렇게 맨날 단어들이 이상하게 용어를 써가지고 무슨. 오케이, 알겠습니다. 그러면 일단 저도 한번 보고, 그리고 그런 방식으로 실제로 수업하는 상황에서는 좀 큰 방에서 지원을 하면 되죠, 뭐. 오케이, 네, 알겠습니다. 네, 네, 감사합니다. 네. 네. All right, guys. So, okay, so no problem. We're basically being asked to follow certain regulations, and these regulations deal with things like, how many students can fit in a room, how far they have to be, that they have to wear masks, and so forth, and so forth. Oh boy. So yeah, things are not really back to normal yet, but let's see how it goes. Okay, guys. So we've talked about all the Operon stuff. So now we're going to move on to biotechnology, and there's a ton of stuff to cover here. So what I'm going to do is, once again, let me just share the screen with you here so we can take a look at a few concepts that I think are really important. And, there's really no one place to start because there's just a lot going on in biotechnology. There's a lot of different concepts that we can talk about here. So why don't I just start off? With a concept that, kind of stems off of DNA replication, and let's see, yeah, and we'll see how it goes. So the first part of biotechnology, the first thing that I want to talk to you guys about is something called PCR, and I think PCR is something that you guys all need to learn how it works. It's a pretty important part of AP Bio. So let me tell you what this is all about. So PCR stands for polymerase chain reaction. So it stands for something called polymerase chain reaction. And the name is actually pretty accurate as to what it really does. So in layman terms, PCR is what you could call a DNA copy machine. Okay. Hold on. Yeah. James 9. What? Hold on, guys. Sorry. I'm just getting a lot of messages. Let me just shoot. Okay, that's fine. All right, sorry about that, guys. You can think of PCR as a DNA copy machine. Okay, DNA copy machine. Now you might wonder, well, why is that important? Why would you need a DNA copy machine? Well, remember, in, let's say, forensic science, which is kind of like CSI, if you guys ever watched CSI, for instance, you have a crime scene and you find, let's say, a hair follicle from a criminal. Now you have this one cell that you get from that criminal, but remember, in a single cell, you have like literally one set of DNA. Now, if you only have that one set of DNA, how many tests can you actually run on that single set of DNA? In fact, in order to run just a regular rudimentary test, you would need many, many, many sets of DNA to figure out exactly what's happening. So without the ability to copy or amplify that DNA, we're going to have some trouble. We're going to have some trouble running any sort of forensics diagnostics test. And there are many other reasons that I'll show you later, but let's figure out how this DNA copy machine works. Now, first and foremost, the person who invented this is a guy by the name of Kary Mullis. Okay, Kary Mullis. And he was a professor of biology or biochemistry. I want to say Stanford University. It might be USC, or it's one of the Californian universities. It's like a really great university, maybe Berkeley or something like that. So he was a professor there, and he's a very interesting fella. He was a playboy. He had a lot of girlfriends. He loved surfing. He was a good-looking guy. And the most interesting thing about Kary Mullis was that he did a lot of drugs. Okay, specifically, he did a lot of LSD. If you don't know what LSD is, this is it's called acid, and it's a psychedelic drug. The thing about psychedelic drugs, though, is that it makes you think beyond, samchawan, like completely changes the way you think about things. So what happened was he was actually driving home after surfing on the California Coast Highway, and he was high on LSD. And he pulls his car over because he has this really great idea, starts jotting down these notes, which would later become the PCR. And of course, this actually wins him the Nobel Prize for biochemistry in the 1980s. So he's high on drugs, comes up with an idea, and then he wins the Nobel Prize. So it's one of those really funny examples in science where you have this psychedelic drug that allowed him to invent something that was very, very useful for us. So let's take a look at what his thought process was first, okay? So he wants to create a DNA copy machine. The thing is, DNA is copied primarily in vivo, which means that in living cells. And in living cells, as you know, as DNA is copied, it creates, sort of a replication fork like this, replication bubble like this, and helicase, polymerase here, and then you have Okazaki fragments, and hopefully you guys remember this, right? There's so many things going on in terms of creating a copy of that DNA that in order to do this in vivo for a DNA that you want to copy, it just wasn't feasible. So he thought, is there a way that we can do this DNA replication in vitro, as in within, let's say, like a test tube, right? Within a test tube, is there a possible way to make DNA copy itself in a test tube? And he kind of took it step by step, right? Because he knew the first thing that you needed to do when you want to copy DNA is that you needed to split the DNA strands into two, right? So let's think about it. Even in vitro, whether we're talking about in vitro, in vivo, the first thing you need to do is to have that DNA split apart like this. That's what you wanted to do, just like that. You want the DNA to split. Now, of course, in vivo, in the cell, this process is done by helicase, right? This is usually done by helicase. But can we do this without helicase, he thought? Maybe we can use something that's a little bit more physical to actually denature DNA. So what is a way to denature DNA? What is the way to separate the DNA strands without a helicase? Heat, right? Heat. So he thought, wait, hold on. I can put the DNA that I want to copy into a test tube, and as long as I raise that temperature up to about 90 degrees Celsius, it's going to actually separate those DNA strands. Now that you have it separated, maybe we can make copies of it, right? So he did that very first step, and that was fine. But the second step is where the problem was. So now that you have these two strands that are separated like this, you needed to have a polymerase that would copy those DNA strands like that, right? You needed a polymerase to do this. But the problem is that in order to maintain this DNA as separate molecules, you need it to be at least around 72 degrees. You need it to be at least around 72 degrees. And the problem with most DNA polymerases is that they are not stable at 72 degrees. For example, a human cell DNA polymerase is going to be at its optimal at like 37 degrees, which is internal body temperature. And that's going to be the case for most endothermic organisms. So 36 degrees is normal, 72 degrees, not normal. So the DNA polymerase that we have in our bodies would never be able to operate at 72 degrees. So he was stuck, except because he was high, he thought of something that was actually quite brilliant, because he started thinking, wait, what about archaea, right? What about archaea? What about extremophiles? Extremophiles. Specifically, what about thermophiles, right? Think about it. A thermophile is a bacterial species or a series of set of bacterial species that live in really hot spring water. And those waters are like 70 to 80 degrees. So for those bacteria, obviously their DNA polymerases must be optimized. They must be optimized for around 70 to 80 degrees. Isn't that true? Yeah.
Mm,
So he started searching for a candidate microbe, and he found one called the Thermus aquaticus. Thermus aquaticus. It's a bacteria that lives in hot water. Now, from the Thermus aquaticus, he was able to extract their DNA polymerase, and he called it the Taq polymerase, right, from Thermus aquaticus. So he argued that yes, we could split DNA by using heat, and in that high heat condition where the DNA remains split, we can use the Taq polymerase to combine dNTPs, or the nucleotides, into the new strand of DNA, making a copy, right? Making a copy. Now, there was one problem though, because even Taq polymerases required primers. So we still needed to put primers in to start the sequence. So that's going to be the last bit of challenge. But by this time in the 1980s, early 1980s, we were able to synthesize, 6, 7, or 8 oligonucleotides, which means they're short strands of nucleotides. We were able to make short strands of RNA. So by solving that problem of making RNA, he was ready to go. So let me show you what's going to happen here. So imagine that in this big test tube, let's say, in this big test tube, you put in a strand of DNA that you want to copy. Now, hopefully, you know some part of the sequence because you need to make the primer for it. So what you're going to do is you're going to first add some stuff. You're going to add some primers that you're going to use. You're going to add some primers. And then you're also going to add dNTPs, because those are the actual nucleoside triphosphates that's going to be used to build a new DNA. And then you're going to add Taq polymerase. Okay, that's it. I mean, you have some buffers and so forth. So what you do is this. You first bring them up to about 90 degrees. And when you do that, this DNA will actually denature, just like that, right? And then you allow them to come back down to about 60 degrees. Now, this isn't exact, but you bring them down to 60 degrees. Now, at that point, DNA doesn't renature, but what happens is that the primers will actually attach at that temperature. Okay, so the primers will attach to the temperature. So let me actually label some of this. So this is the first step here. So this is the adding of substrates, Okay, and this second part right here is going to be called denaturation. And then the third part here is called annealing. Okay, annealing. Now what you do is you have this temperature go up to about 72 degrees. And at 72 degrees, what's going to happen? Well, remember, we have the primers right here attached. We have the primers attached here. We have the Taq polymerase. So let's make Taq polymerase blue. So Taq polymerase is now going to take this, right? Take this primer and make a copy, just like that. Make a copy of that DNA. So now in that one cycle, so this right now, whole thing that you see here is a single cycle. In this single cycle, you're able to take the DNA in a test tube, you're able to split it, you're able to anneal the primers, and then you're able to use that primer to create 2 copies of that DNA. Now, that's one cycle that takes about 10 minutes. Now, here's the thing. All you have to do is to do this one more time. Because if you do this, then the 2 strands will split. And then it will anneal, and then we'll copy. Now you have 4. Now you run it back up to 90 degrees, they'll split again. And then they'll anneal, and then they'll copy. Now you have 8 strands, and then you have 16 strands, and then you have 32, and then you have 64, 128, 256. If you keep doing this, every cycle, you double the number of DNA that you have, given that you have enough primers and enough dNTPs and sufficient Taq polymerase to make these copies. So this is actually incredibly brilliant because it's so simple. You heat up the DNA to denature them. You add some primers, you add polymerases that work in hot water, and then you continue to use what we call thermal cycling to create copies after copies after copies of that DNA. And after about 30 cycles, you have millions and millions of copies of the original DNA that you started off with one with one single strand of DNA or one double-stranded DNA. This is pretty incredible stuff. It's really, really incredible. And this is why, this PCR technology has won the Nobel Prize, or has won Karen Mullis the Nobel Prize. And just to give you an idea of why this is important, we actually use this PCR in our testing for the coronavirus. So let me show you how that works. So the thing about the coronavirus, first of all, the thing about the coronavirus is that it's novel, it's new, but back in December of 2019, the Chinese scientists released the full genomic sequence of the coronavirus, which means that we know exactly what sequence from ATGC, exactly what the entirety of that, or AUGC actually, because this is the RNA virus, full genomic sequence in RNA, what this coronavirus looks like. And with that, what we are able to do is to make primers. We're able to make primers that are very specific to, so primers specific to, primers, that's right, specific to the coronavirus RNA. Now, here's the only problem though. You can tell that because this is RNA, we can't just use a standard DNA polymerase to do so. So the technique that we're going to do is very similar, but it's going to be called Rt-Pcr, which stands for reverse transcriptase PCR. So in this case, we're going to be using another enzyme, which is a reverse transcriptase, as in it uses RNA to make DNA, okay, uses RNA to make DNA, and then we run the regular Taq polymerase alongside with it to run these tests. So here's what we do. We take a swab, so here's someone's mouth. So we take a swab and we get some of their sputum, right? Now we take this out and we put it into a test tube. And after we put it into a test tube, we run it through some chemical assays. And what we're doing is we're trying to extract RNA. So we're able to extract that RNA from the swab that we have. And what we do is we put this into the PCR machine. But in this PCR machine, we have the primers that are very specific to the coronavirus RNA. So if there is any coronavirus RNA within the sample, then those primers will actually attach to the RNA and start copying. But if there's no coronavirus RNA, then those primers are not going to bind, which means that you're not going to get any amplification. So if you get amplification through RT-PCR, that means you have the coronavirus RNA that's in the sample. If you don't get any amplification, that means they don't have any coronavirus RNA. But the neat thing about this is that the dNTPs that we use here, so the nucleoside triphosphates that we use to create the new strands, are actually labeled with fluorescence. In terms of when they actually attach as a double-stranded structure, they will start to fluoresce under UV light. So as soon as we get that amplification happening, as we at real time shine UV light into our samples, you'll get that fluorescence. And when you see that fluorescence, that tells you, boom, that's positive, right? This person in their nasal sample or nasopharynx sample has the coronavirus RNA. So, this is a technology from the 1980s that has been modified and modified and modified to be able to even detect very, very minute nucleic acid presence, okay, within a system. And another thing that you can do with this is that you can literally go to a lake and you can scoop up some water, extract the DNA, okay? And if you're interested in trying to find certain fish, you can run those primers in and amplify that DNA, and you can tell, like, run it through the sequence, say, okay, well, there's carp, there's tuna, there's herring, and there's this. You can even figure out what species of fish exists in a particular pond by using the environmental DNA that these fish are shedding out, because of the fact that we can take a single minute portion of DNA and amplify it to a great extent. So that's the beautiful part of PCR. So it's really, really cool here, okay? Any questions on PCR?
Cool.
Yeah, good. Not too complicated, right?
Okay.
Okay, good. Now let's move on to the next topic, which is transformation. So we're going to talk about transformation now. So let's do that. All right. And I think this is going to take majority of the remaining time here. So let's take this slow because this one's a little bit tricky. We're going to talk about transformation. So what is transformation? So the transformation is part of a toolkit under what we call recombinant technologies. Okay, recombinant technologies, wherein we're able to take genes from one organism and express it in another organism for some purpose, some reason that we might have. So what might that reason be? So for example, have you guys heard of something called insulin? Insulin, right? You've heard of insulin. So
Yep.
insulin is a hormone that allows blood sugar regulation. And as you guys are aware, people with type 1 diabetes, for instance, or what we call juvenile diabetes, they can't produce insulin. So they have to take insulin shots periodically in order to maintain their blood sugar levels. Now, in the past, and you know, the thing is, insulin was actually discovered at my university, where I went to school, which was kind of nice. We still have their like pictures up on the wall because that's the last time we actually did anything useful. Actually, that's not true. But anyway, what they found out was that you can use pig insulin to treat diabetics. And that's what we did. You know, we used to take pigs, get their pancreas, extract their insulin, and then we would make medication for the patients. But there are limitations, right? First of all, it's not human insulin. Okay, so there's that factor. But more importantly, you can't produce enough to meet the demands of the growing population who are diabetic. So what scientists have decided to do was to extract the genes for insulin from the human genome, put it into a bacterial plasmid, and put that plasmid into a bacteria, and then have that bacteria produce a lot of insulin, which we extract later, slap it on in the case, and sell it, right? That's what recombinant technology allows you to do. So let's think about some of these background knowledge that we need to understand how this transformation is going to occur. Now, first of all, as I mentioned, we're going to be using bacteria like E. coli, right? Like E. coli to perform a transformation experiment. Now, of course, in your school lab, you're most likely going to use E. coli because that's the most common and most easily workable bacteria. Now, before we said that E. coli had a chromosome. Now let's make that chromosome, you know, pink here. So let's call it the chromosome. But another thing that we realize within bacteria is that they actually have quite a bit of other small circular DNA that we call plasmids. And these plasmids are almost like miniature chromosomes. And these plasmids can have genetic information about 4,000 to 6,000 base pairs long, and they're able to, you know, be transcribed and translated much like their main chromosome. So these bacteria have this. So what the scientists want to do is this. What if we're to be able to, let's say, I don't know, let's go with the green here. What if we take one of these plasmids out, right? We take one of those plasmids out, and what we'll do is we'll take the human insulin gene. So let's write this here. That's the insulin from human, insulin gene from human. And what if I were to sort of incorporate that into the plasmid, right? Insert that into the plasmid. Now, maybe I should draw the plasmid a little bit bigger so everything is a little bit to scale here. Yeah, what if I were to have a plasmid like this, right? Plasmid like this. I take this insulin gene and I put it into this plasmid right there, and then put this plasmid back in, right? That's the main idea. That's the main idea of how we're going to try to make these recombinant E. coli. Because once this E. coli gets this insulin gene, we just have to trigger certain transcription factors or certain foods to get this E. coli to start producing that insulin en masse starting right away, right? So that's what we want to do. Okay, so let's figure out this first problem. The first problem is that we want to cut out the human insulin gene and we want to insert that into this plasmid. So the first question is, how do we cut DNA and how do we put that into the plasmid? So here's where the scientists are going to be really, really clever, because we're going to talk about a very specific type of proteins that we have within our arsenal, and they're going to be called restriction enzymes. Okay, they're actually called restriction endonucleases, but I'm just going to call them restriction enzymes because that's how they're referred to in most AP biology curriculum. So what are restriction enzymes? Well, these are what we call molecular scissors. They literally act like scissors to cut out these different sequences of DNA. So they cut, okay, so these guys cut DNA at specific sequences that we call recognition sites, Okay, so now I'm going to give you an example of one of these restriction enzymes, and from that we'll talk about where these enzymes come from. So the very first example that I want to refer to is something called EcoR1. So EcoR1 is an example of a restriction enzyme, EcoR1. Now, this EcoR1 happens to cut at GAATTC from 5 to 3 prime. So when it recognizes GAATTC, it's going to cut it. But now, one thing that you'll recognize as well is that this is actually what we call a palindromic sequence. Because take a look at what happens on the opposite strand. From 5 to 3 prime on one strand, it's going to look like GAATTC. Now, when I read it the other way, GAATTC. This is what we call a palindrome. And it is at these palindromic sequences, now, not all palindromes, but palindrome sequences that are very specific to that restriction enzyme that is going to cut this DNA. But the way that EcoR1 cuts, here's something that's even more interesting, is that it cuts like this. Boom, 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 just like that. So it cuts the DNA into these fragments like that. Does that make sense to you guys? Okay. Now, I told you the origin. I told you that we're going to talk about the origin of this. Now, if you take a look at the name EcoR1, this eco actually refers to E. coli. Because the thing is, these restriction enzymes were first discovered in bacteria. So we have things like He3, right? And that's going to be Haemophilus influenzae, you know, and so forth. Like we actually have these restriction enzymes that are named after bacteria. So the question that you have to ask yourself is, why would bacteria create enzymes that cut DNA? And the answer is that they're trying to protect themselves against viruses. Because as you guys know, bacteriophages have been attacking viruses for thousands, if not millions, if not billions of years. Of course, billions of years. And as you know, bacteria simply insert their DNA into the cytoplasm of bacteria. And when bacteria recognize this, they start creating these restriction enzymes, which can then chop up the DNA of viruses. So it's kind of like a chemical weaponry that these bacteria are using. And we look at it and we say, huh, well, that's neat. Maybe we can use it to our benefit, right? And that's how we're using it here. So an example that we're using right now is EcoR1, and it's going to recognize GAATTC palindrome, and it's going to cut the DNA at a very specific pattern like this, right there. So it's like a step ladder pattern. And this is going to help us quite a bit in being able to cut out the human insulin gene and putting it into this plasmid. So let's go ahead and draw this human insulin gene a little bit bigger, right there. Let's draw this a little bit bigger here, and imagine that we have insulin gene which spans maybe all the way from here to here, and somewhere upstream we have something like, I don't know, let's say, maybe I should draw it like that. Maybe I should draw it a little bit differently. Human insulin gene, which is here, and then it keeps going over here, and then maybe over here we have GAATTC here, and of course this would be CTTAAG, something like this. All right, so we have this recognition site here, and let's just kind of like draw it like this, so it continues that way, and then insulin gene here. Let's write, okay, so here's another GAATTC here. Now that of course is going to be CTTAAG here, and let's write it like this. So it continues that way, continues that way. Okay, now the first thing that we're going to do here, as you guys can imagine, is I'm going to digest this length of chromosome with EcoR1, because you can see that I have actually two restriction sites for EcoR1 here. GAATTC here, boom, right there. So it's going to cut like this, that's it, right there. And it's going to cut like this, right there, boom, like that. Okay, so what we have after we cut this insulin gene is that we have a fragment of DNA that looks something like that. Okay, does that make sense to you? And then of course, the other side would have something like this, you know, something like this over here, but you know, that's gonna be thrown out. So who cares really about the remaining part, okay? So that's what we have. And as you can tell, if I cut this out, then what I would end up with, let me make this in red, what I would end up with is something that looks like this. Something that looks like this, right? With the insulin gene in the middle. That's the insulin gene. And over on this side, I have A, T, T, C, and G. And over on this side, I have G, C, T, T, A, right? So that's what I have. As you can tell, it's going to have those little ends that stick out. Does this make sense to you guys so far? Yeah?
Yeah.
Okay, good. Now, again, I can't guarantee that that GAATTC is going to be there, but you know, most likely these are going to be sequences that are fairly common, all right? Now, here's what I'm going to do. So now I was able to cut this out. So that much is done. Now I need to be able to insert that into the plasmid. So here's what I'm going to do. So I take the plasmid, right? So let's say that the plasmid looks something like this. Let's use a different color here. Let's use black and say, okay, here's the plasmid that looks like this. And along this plasmid, I probably have at some point GAATTC as well, right? Because it's a pretty big plasmid. Now, if I also digest this with EcoR1, what happens? Well, let's take a look. I'm also going to cut it like this. Boom, 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 right? And I'm going to get something like this. Yes? Okay, good. So now, let's imagine something here. Let's imagine that I take this little ring and just kind of open it up. Let's kind of open it up this way and open it up this way. You know what I get? I get something like this. I get boom, boom, boom on this side. Oh, you know what? I'm going to actually redraw that because this is bad. Let me actually get rid of that EcoR1. I'm going to write EcoR1 at the bottom here. So I digested this with EcoR1. Okay, now if I start opening this up, then I'm going to have essentially a plasmid that opens up like this on one end, like this, and then opens up like this on the other end. You guys agree with that, right? Because I cut it right here. So I start splitting these apart. I'm going to get AATTCG here, and I'm going to get GCTTAA here. Good, right? So I have this plasmid that's now kind of taken apart, kind of like a keychain ring that you sort of open up. Now, here's what's really cool. If you take a look at what we have, that this portion right here fits perfectly in right there, doesn't it? With TTAA, that's it. And this portion right here fits perfectly right there with AATT. And then if I were to sort of do this, you can see that here will be the insulin gene right there. And what I was able to do is by using that restriction enzyme, cleave off, right, the insulin gene and cut once along the plasmid here and use that information or use that sort of what we call sticky ends, right? These are going to be called sticky ends to combine this insulin gene into the plasmid. Does that make sense to you guys? I mean, it's pretty intuitive, right? So it's really cool how this all works, you know? And, you know, as you guys understand complementary base pairing, this shouldn't be too much of a problem for you. So what we've been able to do then is then we've been able to take a plasmid, a regular plasmid, and we're able to put the insulin gene, right? A recombined insulin gene into this plasmid. And all we have to do then is to put this plasmid back in and voila, we're done. Good? Okay, but it's not that simple. So it's not that simple. So I'm going to tell you why this isn't as simple as simply putting that plasmid in. So let's go over here and actually talk a little about how we actually do this transformation in a lab. So let's talk about transformation protocol. Okay, so transformation. All right, so let's actually draw that plasmid here. So this is the plasmid. And the plasmid is going to have the gene that we want. So let's write insulin here. And this is what we want to put in to a bacteria, which would be much larger than the plasmid itself, right? It's a bacteria that looks like this. All right, great. We're going to put this in here. So how we're going to do this is actually pretty interesting. So first of all, we're going to take we're going to make a lot of this plasmids, right? And we're going to make a lot of plasmid and create a solution. And for this, I'm just going to, draw these little black dots. And these black dots are supposed to represent a bunch of plasmids with insulin on them, okay? And then we're obviously going to have a big test tube. We obviously have a big test tube, and this test tube is going to be filled with bacteria. So let's draw little bacteria here. So these are all going to be E. coli bacteria. Now, there are some other chemicals in here. There's going to be some buffers. There's going to be some, agar solution, solution for the bacteria to eat and so forth. But, it's actually pretty simple, the first step. We're going to put this, actually, let's start off here. We're going to start off by putting this in on ice. Okay, we're going to really cool down. We're going to really cool down those bacterial cells. Now, after we put them into ice, we're going to take these plasmids and we're going to insert them into that solution. So when we do this, you're going to have plasmids that are going to be intermixed in the solution just like so. Does that make sense? Okay, so we have a bunch of plasmid here. Now, what we do after this is that we put them into a 42-degree-ish, about 42-degree heat hot bath, okay? So we're going to put the entire test tube into a warm bath, warm water for about a minute. And what this does is that this makes the membrane all rukong, right? So that it's really easy for these things to go in. And then what we do is we return them to ice. And then we return them to ice again. And what we're hoping for is that this heat shock, the difference between the 42-degree and the ice, will make some of these plasmids shoot into the bacteria like this, like get sucked into the bacteria as a result of the heat shock, therefore transforming these bacteria. Are you guys following me so far?
Mm-hmm.
Okay, good. Now here's the only problem though. Just as I've drawn here in this diagram, transformation is not always going to be very efficient. So if I have 1,000 cells, I might get 5 or 6 cells that actually get transformed. The other cells are not transformed. And that causes a massive problem for us. Because when I take a look at this test tube right here, I can't tell which ones actually have the plasmid and which ones do not have the plasmid. So I need to be able to distinguish them apart in one shape or another. So here's what we're going to do. Let's go back to that plasmid, shall we? Now, instead of just having insulin, what if I were to actually add something else, like maybe another gene that confers some sort of ampicillin resistance or some antibiotic resistance? So in this case, let's say ampicillin resistance. To such an extent that if this plasmid is inside the bacteria, they'll be resistant to a particular antibiotic. Because here's what I can do. I can take these cells, and in order to separate the ones that received the plasmids from the ones that didn't, I really just simply plate this on an LB or liquid broth. Actually, not LB, sorry. This would be agar. So it would be agar, which is just food, plus ampicillin. Because think about it this way. If you look at these cells, let's say that this one right here was transformed, this one right here was transformed, and the one at the bottom here was transformed, right? These guys would actually be able to grow because of the fact that, yes, they have the plasmid, which has the ampicillin resistance. But the other cells, like this one, or this one, or this one, or this one, that didn't get transformed, well, they don't have ampicillin resistance. And because ampicillin is a 항생제, is an antibiotic, they're going to die when we plate them. So the main idea of this ampicillin resistance then is that it's acting as a screen. It's acting as a filter to only allow the ones with this plasmid to grow, therefore guaranteeing that all of these bacteria that I see growing on this plate 100% have my insulin plasmid within their cells, that they have been perfectly transformed. So then what I do after that is to take those guys and we allow them to replicate, right? Allow them to replicate and then start pumping out insulin. That's the whole point of this sort of recombination effect. Is that clear, what we're doing with the ampicillin? Yes?
Yeah.
Okay, good. Now I have to talk to you about the finer details of how we actually run this experiment with the controls that are in place. Because yes, what we're looking at right now, this agar plus ampicillin is what we'll call our experimental plate. But we still have to have some plates that are going to be made in order to act as controls. So let me actually redraw this in a new diagram and tell you what we're going to be doing. So first of all, you have basically a test tube with bacteria, and you have plasmid, right? So this is going to be what we call our plus plasmid tube. But you have to remember that this plus plasmid tube is going through heat shock, right? Okay. What we also have to do is to create another test tube. And this test tube can't get any plasmids, just the bacteria. That's it, just the bacteria. So this is going to be a minus plasmid tube. And this one is still going to undergo the same heat shock because it is in fact a control test tube. Does this make sense? So you're going to go through the same procedure, except you're going to put plasmids into one of them, and you're not going to put any plasmids into the other one, but you're still using the same strain of bacteria. So we have plus plasmid and we have minus plasmid test tubes. All right, now we're going to plate these guys on two different types of plates. So at the top, we're going to use what we call just agar. Now this one here is going to be agar plus ampicillin. Here, we're going to see agar plus ampicillin. And on the last one, it's just going to be agar, just like this, okay? Now, this one's that experimental plate that we were talking about earlier. Why is that? Because as you guys can tell, some of these plasmids are gonna go in, and the ones that grow here are the ones that definitely have that plasmid, because the plasmid confers that ampicillin resistance, which allows them to grow there. Now, let's talk about what these control plates 1, 3, and 4 are doing for us, because this is plate 2. Okay, so 1, 3, and 4. Now, first of all, I'm going to actually draw bacterial growth with this pink. Okay, so this should be pink here, pink here, pink, and pink. What we're going to see in this plate is going to be full growth. They're all going to be growing, just like this. Now, this actually makes sense. Remember, on this plate, there's no ampicillin. So whether you have the plasmid or not, you should just be able to grow. So you're just going to get a really, really nice thick vat of growth of bacteria on that plate 1. Make sense? Okay. Now, in this plate 3, you should see absolutely no growth. Because remember, none of these guys in the negative plasmid conditions received a plasmid with ampicillin resistance. So if they never are capable of even a chance of getting that ampicillin resistance, they're not going to be able to grow in that ampicillin plate, because ampicillin, once again, is a 항생제 and can't grow there. Makes sense, right? Okay. Now the last plate, which is agar, well, this one should just see full growth like this, right? Just like that. Because of course, in this plate, there's no ampicillin, there's no plasmid, but you don't need plasmid if you're just going to grow an agar anyway. So this is what you should see. So in a successful transformation experiment, you should always see full growth on plate 1, and agar plus amp should be spotty with few colonies. You should see no growth on plate 3, and then you should see full growth on plate 4. Now I'm going to really focus on these plate 3 and plate 4 for a moment, because I think these are really important. Firstly, this plate 3 has to be clean. If you get growth on this plate for whatever reason, do you know what that means? This means that these bacteria are capable of growing on ampicillin. And that means that the cells that you began with were already resistant to ampicillin, which means that this experimental plate that you have here, number 2, doesn't mean anything, because some of these colonies are growing not because they got the plasmid, but because some of those cells were already resistant to ampicillin. So it doesn't guarantee you that 100% success of plasmid being in those cells that are growing as colonies in plate 2. That makes sense, right? So that's why you want this to be completely clear so that you can guarantee that, look, these cells were not resistant to ampicillin, which makes our plate 2 meaningful. It makes our plate 2 have the actual, is acting as an actual screen to kill off any bacteria that didn't get transformed. Good. Now, here's the other thing though. When you look at plate 4, and let's say plate 4 is no growth, right? Let's say plate 3 and 4 are no growth, right? That's a big problem. Because what this means is that for some reason, during some process, maybe heat shock or the original cell culture, if you don't have any growth on plate 4, that means the cells were not viable. The bacteria that you used for plate 3 and plate 4 were not viable. They were not alive. Now, why is that bad? Well, because that means that plate 3 isn't empty because of ampicillin. Plate 3 is empty because there were no cells. And if plate 3 is not going to verify the conditions of non-existing ampicillin resistance in the cells, then plate 2 is meaningless. So let me repeat that. If plate 4 is empty, that means there were no cells to begin with in plate 3 or 4. And plate 3 was supposed to be no growth because the cells died due to ampicillin, not because there were no cells. So it makes plate 3 meaningless. And if plate 3 is meaningless, then plate 2 is meaningless. You see what I'm saying? So there's a lot of things that are at play here that need to happen, that need to coincide with each other in order for them to support each of these conditions and and to ensure that what we see is in fact what we're seeing due to the success of the transformation. Is that clear with you guys, what we're looking at? Yes? Okay, good. So this is a general transformation procedure. Now, I'm just going to show you guys something that I've seen on a previous test that I think was pretty fascinating. So what they did was they wanted to mix this idea of transformation with operons. So this is what they said. Now I'm going to just modify this a little bit. And let's say that there was we took Lac Operon, let's say. We took Lac Operon and we put the Lac Operon onto a plasmid. Okay. So let's see what that would look like first. So we have a plasmid here, and in one section of the plasmid, we have the promoter, we have the operator, and then we had the Z, Y, and A. Like that's what that would mean for this. Ooh, this is really ugly, sorry guys, but that's what it would mean, right? We put the Lac Operon, which is this entire thing, onto the plasmid. And let's just make an assumption that they probably were able to put the Lac inhibitor, right? Lac inhibitor and its promoter here onto the thing as well. All right, so now this is what they say. After we put this Lac Operon in, we went in and took out these structural genes. We actually removed those structural genes, and we've actually replaced this with another protein, another gene, sorry, that we call GFP, the green fluorescent protein. Now, green fluorescent protein is a gene that's actually produced in jellyfish that produces these green fluorescent proteins. So under UV radiation, they fluoresce green. All right, and then they say, all right, we put this into a bacteria, right? We put this into bacteria, or we tried to anyway, and we plated these guys. We plated this plus plasmid vial, and we plated them on 4 different conditions. So I want to talk about what these 4 different conditions are and explain why we're seeing what we're seeing. So the first condition here was, I think, just agar. Okay. Oh, and by the way, sorry guys, by the way, there's obviously ampicillin resistance here as well, right? There's obviously AMP-R here as well. Okay. Ampicillin resistance. All right. So on the first plate, we had agar, we had no AMP, and we have no lactose. And what they say is, all right, well, when we look at these guys under normal light, we had lots of colonies that were all white colonies. Okay, they were not glowing green, obviously, But even under UV light, all of these colonies were still white colonies. Does that make sense to you guys? Okay, that's because first of all, these guys have agar, which is fine. There's no ampicillin, so they all grow, and there's no lactose. Now remember, some of those guys could potentially glow green, but if there's no lactose, this GFP is not going to be turned on, right? So that's why they see just white colonies. Now in the second one, we have agar, we have ampicillin, and we have no lactose. And let's see what we see here. So under normal light, what we see here is very few white colonies. Few white colonies. Now let's see why that's the case. First of all, ampicillin is in effect, which means that all the cells that didn't get transformed died. So under normal light, we're going to see very few white colonies. But even under UV light, we're still going to see few white colonies because there's no lactose. And without lactose, we're not turning that GFP on. So the next one that we played it on is agar plus amp and lactose. Now here's something that's interesting, because under normal light, we see the same thing, few white colonies. However, under UV light, what we see is few green colonies. Now remember, in this case, we're seeing green colonies because there's presence of lactose. And the presence of lactose allows these genes to be produced, because remember, if you have lactose, this one falls off, and the promoter binds to RNA polymerase, produces GFP. This is what's glowing under UV light. So under UV light, we have few green colonies that exist. And there's no other colonies. It's just few white colonies that turns green.
But why not in normal light?
In normal light, because remember, fluorescence only happens under UV radiation. That's
okay.
Yeah. So you just need to shine UV light, you know? So that's, yeah, it is what it is. But I think we have one more, right? We want agar, we want no ampicillin, and then we want to put lactose. So let's predict what's going to happen here. In this case, under normal light, you're going to get lots of white colonies. You're going to get lots of white colonies. Under UV, you're going to see mostly white colonies, and then you're going to see few green, right? Because here we're not using amp. So it means that there are going to be both transformed and untransformed bacteria on this plate. But there is lactose. That means the transformed ones are in fact going to glow green under UV. But most of them are not transformed. So we have mostly white and few green. Does that make sense to you guys? So this is sort of a way to combine what we learned about with the operons and how, you know, lactose can regulate the, you know, the production of this GFP in this case, and the presence of lactose can do that. And of course, how that transformation experiment is going to tie in with everything. So I know that this last part was a little rushed, and it's not the most intuitive thing to do over the internet. And I wish I could actually go back into class with you guys right now. It's not the best time, but that does get us through some of the more important transformation stuff here. Now, there are very few additional things that we need to cover. They're going to be things like transduction, conjugation, gel electrophoresis, but I do think those things are fairly easy, and we will talk about them probably in the next class right before we do our open sort of Q&A session like that. But look, let me finish up by saying this. First of all, I will have this recording up on the internet at some point within the next couple of days. So like I said, I feel like I've been able to cover majority of the things that you need to know without having you guys study for like tests and sort of things like that. I know that it's not always easy to sit there and really try to understand what's happening. I know some of you guys can do that, but you know, I know that it wasn't easy for me. If I'm not studying for something for an exam, you know, I just kind of read over it, gloss over it, and that's all. You know, but the thing is, over the next couple of months, we're really going to start looking at these things more closely and trying to test your knowledge on how much you guys remember, how much you guys know, and how much you guys understand to apply these concepts. So here's what I'll say. Next Tuesday, let's have the last class, last lecture on finishing up the phoresis, finishing up some of the biotech stuff, and then addressing whatever questions. And we can have it an hour, 2 hours, whatever you guys want. So long as you guys have questions, we'll try to answer all of them as possible to the best of our ability. And then on the Friday maybe, or the next week, or next Tuesday or something like that, depending on how this whole Corona thing works out, I'm going to have you guys solve the level test, okay? And we're gonna actually set aside some time to talk about the level test, look at the questions and say, look, this is the breadth of knowledge that AP Bio wants you to know. You know, this is what you need to know. And this is how we connect these ideas together, because AP Bio questions are really good. So we're almost ready, guys. So today is March 24th. And I think within 1 week, April 1st, and then maybe the week after that, we're gonna be ready to start practicing the test. That still gives us about 6 weeks. And now we're gonna be doing a lot more focus on FRQ questions. I know that's gonna be tough, but we'll do our best. And my hope is that by the middle of April, that you guys will be back in the classroom. You know, hopefully everything will return to normal. Things are happening over there in the States. Bad things are happening in Europe. Good things are happening here. So hopefully we'll get you back into the classroom. I know there's a tremendous amount of limitation on doing online classes. And I've done about 60, 70 hours of these classes now at this point, just in the last couple of months. And I'm really starting to see this is not as nearly as good as you guys being in the classroom. And you guys know that too. So it's a little disappointing, and I wish that we can, you know, go back into the class faster, but that's something that's out of our control, right? So we'll try to do our best. And I think the point that I want to make is that, again, if you guys don't feel comfortable with the May exam, you can take the later exam, which means that we have that much more time. And I'll be more than happy to work with you guys until you need me, so that we can get ready for this exam. But I feel like we're getting pretty close to the end. Read the textbook, read the sections that you're uncomfortable with. Bring those questions to me next week. I'll remind you guys again, but in the next week, what I want us to do is to have units 1 through 6 all open and say, let's talk about any one of these things that you guys can't explain off the top of your head, right? And talk about it. And that's what we're gonna do, okay? So until that time, guys, look over the units. It's really simple. It's available on the internet. You know, if you go on Google and just type in, let's see, AP Biology, the course, right? It's the second one here, Course at a Glance. So go through this, look at it, read the sections that you don't understand, and then bring your questions to me next Tuesday. Does that sound good?
Cool.
All right, guys. That's it for me today. You guys have a great evening. Stay safe out there, and we'll see you in about a week, okay?
Yeah.
Bye-bye.
Bye.
Bye.
Bye.